Okay po, good morning. Doc, rinig po ba ako?
Yes, rinig ka, Doc. Very clear.
Okay. So, good morning po. Good morning, everyone. Thank you for attending this lecture. I'll try to make this as compact and as concise as possible. So, for today, we'll be tackling general microbiology. So, this will encompass a brief overview of our microorganisms, bacteria, viruses, fungi, etc. Okay. So microbiology is the study of these microorganisms, and this deals with the morphology. So yung sizes nila as well as yung shapes, presentations kapag titingnan natin through the microscope or through our naked eyes. So individually, these microorganisms, they cannot be seen readily, unless they are forming colonies na, especially sa ating mga bacterial species. So for bacteria, when they are being measured by their colonies, we use the units millimeters. However, when they are measured individually, micrometers yung unit na gamit natin. So they may be beneficial or non-beneficial. So they may also be pathogenic or non-pathogenic, opportunistic or non-opportunistic. So some of them, they are being used for fermentation. Some of them are used for vaccine production, etc. Ayan, and opportunistic pathogens, these are microorganisms that produce disease once the animal is immunocompromised. Yan, so microbes present in the living organism, these are called as normal microflora or normal microbiota. And most stark example of this would be, what do you call this? Most stark example of this would be the rumen microflora. So we have cellulase producing microorganisms such as Fibrobacter and Enterobacter species. Yan. Next. So applications of microbiology. So knowledge of this subject would help you or help veterinarians deal with the following. So first one is prevent disease occurrence, which is very important in epidemiological studies. Ayan, so knowing how a disease is being transmitted or ano yung mga magiging signs niya would help you control it, especially yung mga economically important diseases natin such as yung ASF. Ayun. And for treatment of infectious diseases, this is important for medicine naman. So specific microorganisms usually have specific countermeasures against them. Second would be on public health and biosecurity. So prevention of food spoilage and assurance na safe yung mga food natin galing sa slaughterhouses, etc. Next would microbiology is also important in our laboratory and surgical settings. So aseptic techniques, etc. These are developed through the knowledge of microbiology. Ayun. So yung hand washing natin na-develop dati because of knowledge of microbiology, of course. Okay. So before before bago nila ma-develop yung germ theory of disease, bago nila malaman or bago nila ma-discover yung mga microorganisms, diseases were believed to be a product of sins. So iniisip nila makasalanan ka kapag nagkasakit ka dati. And before that, meron din tayong tinatawag na imbalances sa humors. Iniisip nila kapag nagkasakit ka, merong mali doon sa mga fluids na tumatakbo sa katawan mo. So ang ginagawa lang nila dati, instead of using medicines, ay sinusugatan yung mga tao to drain yung mga bad humors na nagsisirculate, etc. So history, skip natin yung iba kasi hindi naman sila masyado lumalabas sa board exams. However, I will throw in information every now and then with regards sa history ng ating microorganisms or microbiology. So initially, Robert Koch and Jacob Henle tried to explain how infectious diseases were developed, and they produced the Koch postulates. So as a review, ito yung mga apat na postulates natin. So a specific microorganism can always be found associated with a certain disease. The microorganism can be isolated and grown in pure culture in the laboratory. The pure culture of the microorganism will produce disease when injected to a susceptible animal, and it's possible to recover the injected microorganism from the experimental animal. Now, sa boards, ang questions na itatanong nila usually dito is, which of the following does not describe the Koch or Koch postulates, or which of the following accurately describes Koch postulates. So it's important to memorize this kahit hindi naman eksakto as long as you have a general idea on anong mga ibig niyang sabihin. Ayan, so Koch postulates, they are not applicable 100% of the time kasi itong mga nakasulat dito, usually bacteria lang ang mga tinatakal niya. So what are certain situations na hindi siya applicable? We have the following. So certain agents, they cannot be grown in vitro, only in vivo. So only on living animals. And examples nito would be the viruses and prions. They cannot be grown in the laboratory, or at least some of them cannot be grown in the laboratory, only sa mga hayop. It's not always possible to find a laboratory animal that can be infected with a pathogen that causes human disease. Kasi there are certain instances in which iba yung magiging clinical manifestation ng isang infection doon sa ating animal as compared doon sa human counterpart niya. Okay? So some diseases are caused by more than one organism. A very good example of this would be kennel cough or infectious canine tracheobronchitis. So there are, I think, more than 5 implicated etiologic agents for that. So we have canine distemper virus, Bordetella bronchiceptica, canine adenovirus 2, etc. And some organisms can cause more than one disease. Yan. For example, we have Clostridium perfringens. Dami yung clinical manifestations doon. Depende sa type. And certain pathogens become altered when grown in vitro. So some of them may become more pathogenic or may become less pathogenic kapag rino nila sa laboratory. Yan. So in light of this, Thomas Rivers developed the Rivers postulates, and this deals specifically with diseases that are caused by viruses. Ayan, so there are two. So a specific virus must be found associated with a disease with a degree of regularity. And dapat yung virus would occur not as an incidental pero as a main cause of the disease in investigation. Okay? So let us classify them as prokaryotic and eukaryotic organisms. So prokaryotic microorganisms, this includes yung bacteria natin. So the main difference between prokaryotic and eukaryotic cells, kapag tinignan nyo sila, would be the presence of membrane-bound organelles. And eukaryotic cells, this includes yung mga fungi natin, algae, protozoa, as well as yung nasa body rin natin na cells. So prokaryotic cells, they are smaller and less complex compared sa mga eukaryotic cells natin. Tapos, yeah, when it comes to the ribosomes, ito yung mga measurements nila. So for prokaryotic cells, we have 70S, and for eukaryotic cells, we have 80S ribosomes. So S, this refers to your Svedberg units. Hindi ko alam kung tama yung pag-pronounce ko, pero spell na lang natin. So SVED, BERG units. And this measures yung sedimentation rate ng ribosomes natin. 80S is considered as the complete or the more complete Svedberg unit sa kanila. Ayan, so prokaryotic cells, they do not have a nucleus, rather nucleoid yung presence sa kanila. However, they also contain chromosomes. Ayan. Nuclear membrane is absent for prokaryotic cells. For eukaryotic cells, it is present. And prokaryotic cells, they replicate or they multiply via binary fission. This is a asexual form of reproduction. And eukaryotic cells, they replicate by mitosis. Okay. So for prokaryotic cells, here are the structures na kailangan natin i-take note or mga important structures. So first one would be the cell wall. It's very important for classification as well as bacterial control. So the bacterial cell wall contains peptidoglycans. So regardless if it is gram-positive or gram-negative, peptidoglycan is present. Amount lang ang nagdi-differ. So for gram-positive cells, peptidoglycan is more abundant as compared to your gram-negative cells. Gram-negative cell wall contains more lipopolysaccharide. And yun na rin yung basis sa kanilang endotoxin production. Okay, so peptidoglycan provides strength and rigidity to the cell wall and contains the following: N-acetylmuramic acid or NAM, tapos NAG, or N-acetylglucosan. Ayun. So, Gram staining, this was developed by Hans Christian Gram. And this classifies a bacteria if it is gram-positive or gram-negative. Bali, ang basis dyan would be yung CVI complex natin. So for gram-positive bacteria, after staining, yung CVR, yung crystal violet and yung iodine natin, they bind within the cell wall. And for gram-positive bacteria, natatrap sila sa loob due to the abundant peptidoglycan content. So, hindi gumagana yung counter stain natin. However, for gram-negative cells, manipis ang kanilang peptidoglycan content. So, after denaturation with alcohol, nawawash off din yung crystal violet iodine complex. And doon na pumapasok yung safranin na counter stain natin. Which explains why yung gram-positive cells natin stain blue and gram-negative cells stain red or pinkish after gram staining. Ayan. Point of comparison lang, sinamarize lang natin. Excuse me. Next, we have the glycocalyx. This is a gelatinous polymer produced by the cell membrane and a component of biofilms. So, when you are looking at stagnant water, kung makikita nyo yung parang sebo doon sa tuktok niya, those are biofilms. And glycocalyx lang yun ng mga bacteria na present doon sa tubig. So, we have two types, a capsule and slime layer. Capsule is highly organized and dense siya. Yung arrangement nila would be tight as compared to the slime layer na irregular siya and hindi masyadong makapal yung arrangement. Slime layer, ano siya? These are found sa plaque, sa ngipin. Yung, yung namumuo dyan kapag hindi natin nalilinis. Those are slime layers. Okay, what are the functions of the glycocalyx? First and foremost, for bacterial adherence to host surfaces. They may also serve as protection from desiccation as well as phagocytosis. They may also act as food reservoir and prevents lysis ng bacterial cell. Next, appendages. This may be divided into appendages for motility or attachment. So for motility, we have flagella and axial filament. Flagella is found in most bacteria and these are thin and hair-like. Axial filament, this is specifically found in your spirochetes, such as Leptospira species. For attachment, we have pili and fimbria. So pili is hollow with the flagella but are non-helical and may be involved in sexual reproduction. So certain bacterial species, especially gram-negative bacteria, meron silang sexual reproduction and this is known as conjugation. So this facilitates information or DNA exchange via sex pilus. Ayun. Meron din naman sa gram-positive pero very few lang sila. Very few lang yung gumagawa ng ganun. So bacteria, they may be grouped based on the arrangement of their flagella. So Atricus, these are bacteria that has no flagella and therefore hindi sila nakakagalaw. They are non-motile. Monotrichus has a single flagellum, similar to your sperm cells. Lophotrichus, group of flagella at one pole. Amphitrichus, flagellum on both poles, single flagellum on both poles. Peritrichus, flagella all over the cell. Ang ginagawa ko dito na palatandaan for peritrichus would be peri, yung suffix na peri. So, niisip ko na nasa perimeter siya ng bacterial cell. So, all over. Then, Amphitrichus, top of flagella on both poles. Next, we have the internal structures. So, chromosome, gaya ng sabi natin kanina, this is stored sa nucleoid instead of nucleus. Hindi siya membrane-bound. Then, we have plasmids. They encode genetic information. Ribosomes, these are involved in amino acid synthesis, storage granules, gas vesicles, and then we have endospores, which is the dormant stage of a bacterial species. So there are only two bacterial species na nagpo-produce ng endospores, which is of veterinary importance. We have Bacillus and Clostridium. Yeah. So more on endospores. So sporogenesis would be the process of endospore formation. While they are in endospore form, bacterial cells have no metabolic activity. Also take note, magkaiba si Bacillus and Clostridium. Bacillus is aerobic. On the other hand, Clostridium is anaerobic. strict aerobe and strict anaerobe. So endospores are resistant because of the following. Because of, first and foremost, yung layered structure nila and high content of dipicolinic or diaminopicolinic acid. Nagvavary kung ano yung sinasabi dito sa iba-ibang sources. However, if you can see this sa mga choices sa board exam, just choose one kung ano yung pinakamalapit. Kasi ganun usually. Kung walang eksakto, choose what is the nearest answer doon sa choices nila. At times, makakakita rin kayo ng wrong spelling doon sa mga choices. Pero yun nga, choose lang kung ano yung mga pinakamalapit. Tapos yun. And sports, wala ulit metabolic activity. And for the production, dapat merong calcium. So, calcium-rich soils, mas maraming bacterial species na present. Mas maraming endospores na present. Okay? Next, we have the major groups of microorganisms. Excuse me. So first, protozoa. Although protozoa, I think, is considered as a parasite. No. So more on this sa inyong parasitology, introduction lang tayo dito. They are eukaryotic and single-celled, and they can swim. Majority of them are motile, and majority of them are free-living. Wala silang kinokos na disease. I think less than 10% of protozoa present sa environment ang pathogenic lang. 90% pataas, free-living. Ayan. They lack cell walls. Yun lang. They have two forms, cyst, which is the resting form, and the trophozoite, which is your vegetative form. So, skip natin. Then next, we have algae. So in recent years, uh, may isang particular na question regarding algae na lumalabas sa board exam, and this involves yung blue-green algae. So algae are plant-like, have rigid cell walls, maybe unicellular or multicellular, and grow in many different environments. So more on the blue-green algae. Ang question na lumabas sa amin nung time na yun is, nagtatanong siya, merong aso daw na nag-swimming sa isang river, sa isang lake. Tapos ang nangyari is, after several days daw, nagka-develop siya ng icterus as well as vomiting, diarrhea, and seizures. Tinanong nila kung ano yung pinaka-possible na diagnosis. So, blue-green algae. Nasa Merck's yun. Pwede nyo siyang basahin for more in-depth information. Pero blue-green algae, these are also known as cyanobacteria. Ayun, so cyanobacteria, they produce several types of toxins, and usually nakukuha natin or nakukuha sila ng animal species via ingestion. Ayun. So other than that, magkakaroon din ng skin irritation after several days kapag naglangoy sila sa areas with blue-green algae. Ayun. So they also occur in colonies. Okay, next we have fungi. So they are eukaryotic. They also have cell walls and maybe unicellular or multicellular. Ginagawa kong palatandaan ulit dito for multicellular forms of fungi would be M to M. So multicellular, they are molds, and unicellular, these are yeasts. They produce via spores. They may be micro or macroscopic in size. So similar to our bacteria, individually hindi nyo sila makikita since they are very small. However, once they reach sufficient numbers and magpo-form na sila ng colonies, pwede nyo nang makita and mahawakan to your naked eyes or hands. Ayan. So anatomic considerations, mas marami compared to your bacterial species. So they produce filaments. So hypha then magpo-form ng mycelium. Ayan. So what is the main difference from bacteria? Instead of peptidoglycan, fungal cell walls contain chitin. Ayan. So the cell walls also contain plenty of ergosterol rather than cholesterol, and this is the target of our antifungal drugs, yung ergosterol nila. Ayan. So bacteria, they require more water as compared to your fungal species. So classifications of fungi. Excuse me. First, class Phycomycetes. These are bread molds and leaf molds. Pag titignan nyo under the microscope, ang pinaka-common dito would be Rhizopus species. Then we have Ascomycetes, the sac fungi. These are your cheese molds and mildews. Palatandaan dito would be ASC. Kasi yun ang spelling ng sac. Yun na. So Ascomycetes. Next, we have Basidiomycetes. These are the mushrooms, toads, tools, rusts, and smuts. In veterinary medicine, only a single species is of veterinary importance, I think. It's Amanita species. Nasa marks din siya. Pwede niyong basahin kasi lumalabas din siya usually sa boards. It's either ilalagay nila dito sa microbiology and public health or ilalagay nila sa pharmacology and toxicology. Then next we have the class Deuteromycet. These are fungi imperfecti. And kadamihan ng pathogenic fungi na nakikita natin in practice belong to this class. Next we have bacteria. They are prokaryotic species. And marami siyang classifications. However, there's only two broad classification of bacteria. We have Eubacteria and Archaeobacteria. So, Eubacteria, these are also known as the true bacteria, and majority of pathogenic and non-pathogenic species of importance belong there. Eubacteria, yan. Ancient bacteria or Archaeobacteria are found in the most extremes of conditions. So, makikita natin sila sa mga bulkan, sa Arctic areas, kung saan it's either napakataas or napakababa ng mga temperature. Okay. So, morphology. Na-mention na natin kanina yung size ng bacteria. Ulitin lang. So, millimeters kapag colonies na ang pinag-uusapan and micrometers kapag individual bacterial species. So, shapes. So, spherical or cocci. Ayan. Then, cylindrical or rod-like for bacilli. Also take note, several bacterial species are pleomorphic or they may have two forms. For example, we have Corynebacterium. Corynebacterium has two forms. Pwede siyang maging club shape or rod shape. Then we have Rhodococcus species. So Rhodococcus, rod shape and spherical shape. Nasa name naman siya. Rod and coccus, Rhodococcus. Then pleomorphic bacteria, these have variety of shapes. Pinakamagandang example for our pleomorphic bacteria would be Mycoplasma species because they lack cell walls. Maraming cholesterol yung ano nila, yung structure nila. Very elastic and pwede sila mag-change ng shape nila. Depende sa kung anong need. Then we have spirochetes. Ayun. They may look like comma or corkscrew. Hindi naman sila masyadong mahirap i-differentiate. So yan. Just a picture of our bacterial species. So arrangements. For cocci, we have the following. Diplococci, dalawang bilog lang siya. Joined together. So bacterial species, kapag ganito ang arrangement, plasmodesma yung nagko-connect sa kanila. Plasmodesma between bacterial cells. Then we have tetrads. So as the name implies, apat sila. Then sarcina, 8. Staphylococcus, bunch of grapes. Pinaka-favorite to kasi madaling tandaan. Staphylococcus aureus, bunch of grapes, tapos color gold. Then Streptococcus chain type. Yun. So take note of your prefixes. So strep, this refers to chain-like arrangements. So streptobacilli, chain-like arrangements, na rod shape. Ayan. More of them. Yeah. As mentioned earlier, tingnan niyo yung Corynebacteria. Pwede siya maging club shape. Pwede maging cocoid. Next, we have bacilli. So, rod-shaped bacteria, streptobacilli, chains, palisades, parang matchsticks naman yung kanilang arrangement. So, okay. So, growth requirements for our bacterial species. So, hindi ko na in-include dito, pero elemental requirements ng mga bacteria natin is similar to all living organisms. So, they require carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. John Spee. Ayan. So first and foremost, they require yung temperature natin. So they are classified based on their temperature requirements, psychrophiles, mesophiles, and thermophiles. So psychrophiles, ang kanilang optimal growth temperature would be around 15 to 20 degrees Celsius, although they can grow din naman sa mas malamig pang environment such as the refrigerator temperature. Ayan, so sir, refrigerator temperature is around, I think, 2 to 8 degrees Celsius. Yun. So they will grow naman. However, if they are not at their optimal temperatures, magiging mas mababa yung kanilang growth rate. Yan. Kapag mas mataas sa kanila sa optimal, they start to die kasi nagkakaroon na ng coagulation ng bacterial proteins. Ayun. Psychrophiles, Pseudomonas, Flavobacterium, Acalagenes. Proteus species, they are also important dito. Pa-include na lang din. And then we have mesophiles, moderate temperature loving microorganisms. So this includes the majority of our pathogens, and pag nagkakaroon tayo ng fever, doon sila nag-start na mamatay kasi bumababa na rin yung growth rate nila. Then thermophiles, heat-loving microorganisms, usually archaeobacteria lang naman tumamatay. So no eukaryotic cells can grow beyond 60 degrees Celsius. However, archaeobacteria pwede. Eto. So please take note sa kanilang minimum growth temperatures, pwedeng lumabas tong mga to. And sa kanilang maximum growth temperatures. Next, we have atmospheric requirements or yung oxygen requirements nila. So different microorganisms or different bacteria, they require different amounts of gases. Ayun. Oxygen is the most important gas here, although carbon dioxide, this is utilized by some bacteria. And gumaganda yung kanilang growth if carbon dioxide is present. So classification based on oxygen. So first we have aerobic or strict aerobes. They require the presence of oxygen to grow. And as mentioned earlier, Bacillus species belongs to aerobic microorganisms. Pati Mycobacterium, they have an affinity or they have a cell tropism for ciliated respiratory epithelium. So, you can guess naman kung anong klaseng disease ang pinoproduce ng ating Mycobacterium species based on their tissue tropism alone. So, respiratory diseases. Next, we have facultative anaerobes. They can use oxygen when present. And this includes majority of our bacterial species. If you will read our manuals later, you will notice na around, I think, 80 to 85% ng mga andun is facultative anaerobes. The next, anaerobic microorganisms, they cannot grow in the presence of oxygen. So, Clostridium. The next, microaerophiles. They are damaged by normal atmospheric oxygen and grow at levels of 1 to 15%. Normal atmospheric oxygen, I think, is around 20%. So 1 to 15% lang yung requirement dito for certain microorganisms. Then capnophiles to carbon dioxide and oxygen to grow. I think a member of this would be Taylorella species. Next, pH. So for our bacteria, they are considered as neutrophiles. Fungi and algae, they are acidophiles. Ayun. So Samurats dextrose agar, usually yung ginagamit siya for fungal cultivation, mataas yung acid content niya. So hindi siya nag-grow ng bacteria. Yan. So osmotic pressure. Uh, so osmotic pressure is defined as the movement of water through membranes. And yung movement niya would be from an area of lesser solute concentration to an area of greater solute concentration. Ayan. So bacteria, they require an isotonic solution, which is the concentration of water or solutes inside and outside the cell is equal. Kasi kapag hindi equal yan, the pressure will attempt to correct yung difference between membranes or between the cells. Then hypertonic solution, concentration of solutes are greater outside the cell. Ayan. So what happens if a bacteria is in a hypertonic solution? Kung saan mas mataas yung solute sa labas. Of course, water will move out and this will lead to shrinking of the bacterial cell. What about hypotonic solution in which the concentration of solutes are greater inside the bacterial cell as compared sa outside? When that happens, the water will move inside the cell and this will cause the bacterial cell to burst. So for hypertonic solutions, ang magiging consequence natin dito would be plasmolysis or shrinking of the bacterial cell. For hypotonic solution naman, ang magiging consequence natin dito would be plasmoptosis or cell bursting. Ideal isotonic solution. Next, we have salty environments. We have only two types, halophilic and haloduric. So halophilic microorganisms or bacterial cells, they thrive in the presence of salt. So example would be Vibrio cholerae. Excuse me. Vibrio cholerae is important for humans because this causes cholera. Haloduric, these are microbes that don't prefer to live in salty environments, however, can survive in them. And an example would be Staphylococcus aureus. Okay. So growth. So generally speaking, growth refers to the increase in the number or size of cells. So in pathology, we have hypertrophy and hyperplasia for growth. However, for microbiology, growth refers to an increase in the total number of cells. Ayun. Tapos, in terms of death, kapag sinabi nating bacterial death, this refers to the absence or inability to reproduce. So, a bacterial population is considered dead or dying kapag hindi na sila nagkakaroon ng binary fission. And so, the average time pala for our binary fission for majority of bacterial species is 20 minutes. Yeah, tapos, the required number of cells, especially for most bacterial species to cause infection is around 1 times 10 raised to 6. Ayan. So binary fission, this is already done. This is similar to mitosis. Pero ano lang siya, nangyayari siya for bacteria. Spore formation is for fungi naman. Ayun. So these are the phases of bacterial growth or the bacterial growth curve. So we have four: the log phase, logarithmic or exponential phase, stationary phase, and then death phase. So log phase, the bacteria still adjusting to their new environment or to their media. So very minimal yung growth na nangyayari dito for our log phase. So log phase, this is important in food preservation. And what we are aiming to do in food preservation is to lengthen or pahabain yung log phase nila. Logarithmic phase, this refers to an exponential increase in the number of bacterial cells. And this is important for antibiotics naman, for administration of antibiotics. For example, uh, if hindi nag-grow yung bacteria, hindi sila nagkakaroon ng cell wall synthesis. And cell wall synthesis is important for beta-lactam antibiotics kasi ini-inhibit nila yung formation ng cell walls. So kapag walang nangyayaring growth or reproduction, walang nangyayaring cell wall synthesis, walang i-inhibit yung antibiotics natin sa mga antibiotics, amoxicillin and cephalosporins. So wala silang mapapatay na bacteria. So if you use uh, antibiotics which inhibit the growth of microorganisms along with those that are inhibiting cellular processes, magkakaroon ka ng antagonistic reaction. Yan. Next, we have stationary phase. Death and growth is equal. So, stable na yung population ng mga bacterial cells natin. Then we have death phase. Growth rate is negative. Hindi na nakakapag-reproduce. So, what happens here is that nagkakaroon na ng accumulation of waste products. Then, tawag dito, nauubusan na sila ng nutrients doon sa environment kaya hindi na sila nakakapagparami. Ayan. Continuous cell lines pwede namang gawin for our bacterial species as long as mag-supplement ka ng nutrients required by the microorganism and mag-alis ka ng waste products. So you can prolong their stationary phase as long as those two requirements are met. So measures of population growth, microscopic count. Itong dalawa na lang ang explain ko since these are simple. So microscopic count, bibilangin nyo lang yung kanilang cells as well as colony count. Then turbidity, this is important for nutrient broths. Titingnan nyo lang if magkakaroon ng cloudy na formation doon sa media and if meron siyang amoy panis. Next, we have the viruses. So, ilan na natapos natin? 1, 2, 3, 4. I think this is the last one na. For, ano lang, for the divisions of our microorganisms. So, this is the simplest and smallest of all infectious agents. And other sources, they don't consider viruses essentially as living organisms kasi wala silang sariling metabolic processes. And central core lang siya ng nucleic acid surrounded by a protein core. So viruses, they can only contain one nucleic acid. It's either RNA or DNA. Hindi sila pwede mag-exist both at the same time. So yun. So nucleocapsid, this is the infective unit of our viruses. So this contains yung nucleic acid core nila and capsid. Ayan. Viruses pala, they may be envelope or non-envelope. And non-envelope viruses are more resistant as compared to the envelope viruses. Kasi yung envelope is more of a liability instead of protection for viral units. So yun, properties or what are the components ng ating viruses? We have nucleic acid and this carries the genetic information. Then we have viral protein. They may contain enzymes aside from their protein coats. Then lipids and carbohydrates. Also, viral spikes, these are known as peplomeres and they help with attachment ng viruses natin. So viral replication involves the following phases or the following processes. First one is attachment or adsorption. Kakapit lang sila sa host surface. It's either external or internal surface. Ayun. Next, we have penetration. So this occurs via fusion of the host cell membrane in the viral envelope. So our viruses, they hijack lang yung host machinery instead of doing things for themselves. Yeah. Next, we have biosynthesis of viral components. Yun, so proteins na, nagkakaroon na ng transcription and translation, your central dogma, nangyayari dito sa biosynthesis. Then next, we have maturation and assembly. Then release. So naked virions, they are released in a burst-like fashion, and envelope virions, they are released through budding. Ito, parang ganito yung magiging process natin. So magbabind sila, then fusion with the host cell membrane. Then magkakaroon na ng central dogma, yung transcription and translation for viral amino acids. Magkakaroon na ng budding or simply release. Then magkatapos na. Dadami na sila ng dadami, then makaka-observe na tayo ng changes doon sa host cell natin. So periods of viral replication similar to our bacterial growth curve, meron din sila. So eclipse period, this occurs before viral maturation and assembly. This is also known as, or other sources indicate as the incubation period kasi wala pa tayong makikitang infections, wala pa tayong makikitang clinical signs, wala pang cytopathic effects, wala pa. Then latent period, dito pataas pa lang yung viral population natin. So this is similar to our lag phase. Lag, parang secondary lag phase. Then rise period, similar to our logarithmic phase. Then burst size, dyan na. Dyan na tayo makaka-observe ng infections or clinical signs. Okay? Next, cultivation of microorganisms. How are we going to demonstrate them sa mga culture media, etc.? So for bacterial species, we are using media. It's either itong mga to. So first one are solid media. So substances mixed in agar, gelatin, or albumin. Ayun. So for solid media, this incorporates the use of carrageenan. And nakukuha siya dun sa mga seaweeds. Baka tanungin lang sa board exam. Ayun. So concentration, pati to, please memorize or take note of this. 3% gelatin. For agar, 2 to 3% gelatin, 12 to 15%. Ayan. Next, we have semi-solid media. These are usually used for demonstration ng bacterial motility. So agar concentration is lower as compared to solid media, 0.5%. Next, we have liquid media. Wala siyang albumin or agar. These are water-based and includes broths, milks, infusions. Bacterial growth is indicated by the production of smell and the production of turbidity. Yeah. So what you see here, wala pa siyang bacteriang nag-grow. Possible nag-incubate pa lang or lalagyan pa lang siya kasi wala pa tayong makikitang cloudiness na present. Next, another classification for our media. So yung isa is yung form lang and this one is for function. So general purpose media, these are simple media, these are not enriched and they exist for demonstration purposes only. They can grow a wide variety of microbes as long as hindi fastidious or hindi sila nagre-require ng blood. So yun, this includes nutrient agar, nutrient broth, brain heart infusion, and TSA. Next, we have selective media. So they are designed to suppress the growth of unwanted media. So please take note, a selective media may also be a differential media, and media may also be a selective media. Ayun, so several types of selective media or several examples. We have subroots dextrose. This only supports the growth of our fungal species. Maconchi agar, Enterobacteria naman yung sinusupport niya yung growth. Mannitol salt agar, I think Staphylococcus species. Ayun, differential media allows you to distinguish between different groups of bacteria and permit tentative identification. So again, sa Maconchi agar tayo, di ba selective siya? I think Enterobacteria kasi they can tolerate the presence of bile kasi Maconchi agar, this contains bile and crystal violet. This is also known as differential because this differentiates the growth of our lactose fermenters and our non-lactose fermenters. Then enriched media. Enriched media, usually this is not highly selective, although selective siya since yung presence lang or nag-grow lang niya yung mga nagre-require ng blood. This has no inhibitory agents, and examples would be blood agar and chocolate agar. So a difference between these two, blood agar contains RBC, red blood cells na walang kahit na anong special process. Then we have chocolate agar which contains powdered hemoglobin. So yung pinaka-difference nila. Ayan, blood agar is a bit redder as compared to chocolate agar. Kasi hemoglobin na nadiran sa chocolate. So, then common media used in veterinary medicine. So first, nutrient agar, very basic. So no need for this. Then we have blood agar, maconchi, selenite broth, Rapoport bacilladis, used for isolation of salmonella. Edwards medium, this is used for isolation of streptococci. This is, I think, the most selective for streptococci. Then next, we have chocolate agar, supplies factor 10 and factor 5. NAD and hemine for Haemophilus species. Brilliant grit agar for Salmonella ulit. XLD agar, Salmonella again. So this turns yellow for lactose fermenters and purple for non-lactose fermenters. EMB agar for E. coli species. Ito siya, si E. coli. So this produces yung characteristic na metallic sheen natin for EMB. Next, Sabrous dextrose or potato agar. This is acidic and supports the growth of fungi. Esculin differentiates streptococci from enterococci DNAs for coagulase positive staphylococcus from coagulase negative. Coagulase pala is important in the pathogenic status of staphylococcus species. If it is present, they are highly pathogenic. If hindi, mas mababa. Then we have selective serum agar for Brucella, Smith Baskerville for Bordetella, Skiro for Campylobacter and Brucella. What you see on the screen is Stonebrings or Lowenstein-Jansen. This is for Mycobacterium species. Twin 80 or Fletcher media for Leptospira. Then Hectoid enteric differentiates Salmonella and other lactose fermenters and non-lactose fermenters. Although Maconchi pa rin yung pinipili natin dito. Then birdseed agar for Cryptococcus neoformans and other. Next, we have viral cultivation. So viral cultivation, this may be performed in three main ways, as you can see on the screen. So pwede siyang tissue culture, pwede siyang live animal, or pwedeng embryonated duck or chicken eggs. So first one, we have living animals. This is a good diagnostic tool for viral pathogenicity. And some bacteria can only grow on, some viruses can only grow on living animals. This is not the method of choice minsan kasi this is very labor extensive and expensive siya. Tapos, usually namamatay na lang yung animal after inoculation. So, wala tayong ma-observe masyadong changes. Then next, we have embryonated eggs. So, embryonated eggs, this can accommodate a wide variety of viruses and depende sa virus, meron silang kanya-kanyang areas kung saan sila ilalagay. You can also use embryos of different ages. to cultivate different types of viruses. Yeah. For example, we have the chorioallantoic membrane, and this will support the growth and occupancy changes. If herpes viruses, smallpox, Eastern equine encephalitis virus ang gagamitin natin. Amniotic cavity, influenza, and mumps, mga pwedeng viruses na mag-grow dyan. Yeah. So this is a summary kung saan sila i-inoculate, tapos kung saan kayo makakita ng changes or cytopathic effects. Which brings me to our next na slide, cytopathic effect. So cytopathic effect, these are localized changes na ma-observe after viral species have replicated. So for our embryonated eggs, these are summarized changes. So embryonic death, then hemorrhage of subcutaneous tissues, congestion of vessels, stunting, tacking of embryo, decreased amniotic fluid, plantoic fluid, increase, then leukocyte infiltration and necrosis. These are non-specific. Some of them are non-specific. However, ang pinaka-specific lang na cytopathic effect would be inclusion bodies. So, different viral species, iba sa kanila merong very specific na inclusion body formation. For example, this one. Ayan. Pero hindi na ito embryonated egg. Tissue culture, so ganun din, magpo-produce ng cytopathic effect. What you see here na inclusion body natin is known as the Negri bodies, and this is often manifested sa cytoplasm ng ating tissue culture. Usually, yung ginagamit nila would be yung brainstem, mga nasa around medulla oblongata area. Ayan. This is pathognomonic, or this is highly specific for rabies, itong mga Negri bodies natin. Ito tong mga black areas na to. Hence the name Negro. Hindi po tayo racist. The next, we have demonstration of our microorganisms. Karamihan ng ginagawa natin dito, especially for bacterial species, would be staining methods. Pinaka-common is yung gram stain. It differentiates gram-positive and gram-negative bacteria. Please take note doon sa time required per stain. Tapos, please take note doon sa function ng mga stains na present. So, crystal violet would be our primary stain. Iodine is the mordant or binder. Alcohol is our decolorizer, and safranin is the counterstain. So reminder lang, gram staining, ang principle nito would be yung crystal violet iodine complex natin. So repeat lang natin. For gram-positive bacteria, the crystal violet and iodine complex, these are trapped sa loob ng gram-positive cell wall due to the presence of high peptidoglycan content. So after washing with alcohol, maalis lang yung konting peptidoglycan sa cell wall. However, the primary stain and the mordant would remain inside the bacterial cell wall. Ayan. However, for gram-negative bacteria, since very minimal yung kanilang peptidoglycan content, after using your alcohol or after using your decolorizer, you will wash away your primary stain, your mordant, as well as the peptidoglycan layer na present. So after staining with your counter stain, ito lang yung makikita nyo yung safranin. For gram-positive bacteria, hindi na papasok sa safranin since the cell wall still contains the crystal violet and the iodine complex, or the CVI complex. Then we have Zell-Nielsen stain. This is used for certain bacterial species, especially those na hindi masyadong maganda kanilang gram staining. Specifically, we have Mycobacterium. Yeah. So Mycobacterium, this stains positive dito sa Zell-Nielsen due to the presence of mycolic acid sa kanilang cell walls. Although may peptidoglycan naman, very minimal din, hindi siya highly specific. Kaya ito na lang yung ginagamit natin. So, bacterial species which stain positive for Zell-Nielsen or modified Zell-Nielsen are known as acid-fast. Ayan. And they will be red kapag positive as compared doon sa kabila. Huwag po tayong malito. Ayan. Next, we have the IMVIC. This is a biochemical reaction naman. And this is used for the differentiation of lactose fermenters. And highly specific siya for our E. coli. So different lactose fermenters, Sa ating mga enterobacteria, iba-ibang kanilang IMVIC reaction. So, IMVIC, this is indole production, methyl red, Voges-Proskauer, and citrate utilization. Please take note sa E. coli, positive, positive, negative, negative yung magiging result dito. Ayan. This is a summary for other enterobacteria. Pwede natin i-match na lang later kung ano yung mga magte-test positive and negative for IMVIC. So, ito ulit si E. coli. Indole, positive. Methyl red, positive. Voges-Proskauer, negative. And citrate, negative. Positive, positive, negative, negative. So, for Proteus, for example, we have positive, negative, negative, negative. Ayun. Okay. Finally, we have our microbial control. So, this may be done via physical or chemical means. And after this, dito na tayo gumagamit ng drugs. Pero start muna tayo dito sa mga mas simple. So, physical means of microbial control, first one is heat. This may be moist or dry. So, moist heat, this consists of boiling and our autoclave, yung mga pinaka-favorite natin. So, for autoclave, ang ginagamit na temperature doon would be 121 degrees Celsius for 15 minutes at 2 to 8 atm or 2 atmospheres or 15 psi yung pressure niya. And this effectively kills everything, pati mga endospores. Boiling naman 100 degrees Celsius, and usually this will kill majority of bacterial species in around 15 minutes then. 15 to 30 minutes. However, meron tayong process na tinatawag na tyndalization, which involves repeated boiling para mamatay lahat, pati yung mga bacterial endospores natin. Ayun. Dry heat, this involves incineration as well as the use of ovens. A rule of thumb pala when using heat in microbial control, every time you increase the temperature by 10 degrees Celsius, nababawasan ng kalahati yung kanilang time required to sterilize a media or sterilize a material. Next, we have low temperatures or lyophilization or freezing. So freezing, this will allow the formation of icicles or crystals doon sa bacterial cell, effectively killing them. Next, we have desiccation. This is important in food preservation and may involve drying or the use of salt. So drying, ibibilad nyo lang siya. And this will dehydrate the bacteria, effectively killing and controlling them. The use of salt naman will absorb yung, excuse me. The use of salt will absorb water from bacterial cells, leading to plasmolysis. The next radiation, this is used for food packaging usually. And filtration, physical means lang naman siya. And this is not really effective, especially kung individual cells pa rin yung ginagamit na or kinokontrol natin. Next, we have the control of bacteria using chemicals. And as you can see here, but majority of chemicals, they have several processes, although I'll try to be brief with our chemicals that are used. So first, we have alcohols. Ito na lang, alcohol. Ethyl alcohol, usually, ang ginagawa nila would be protein denaturation. And they are effective at 70% concentration above. Common alcohols used in veterinary medicine, we have ethyl and isopropyl alcohols. Then next, we have aldehydes, glutaraldehyde, and formaldehyde. So they are very effective in controlling the population of bacteria, pati mga bacterial endospores, kaya nila patayin lahat yan. However, they are carcinogenic, especially formaldehyde. So formalin, this is formaldehyde in liquid form lang naman. Ayan. Then next, we have biguanides, and the most important member here would be chlorhexidine. Siya usually yung ginagamit natin to disinfect our hands and disinfect our tools during surgery. However, the weakness of chlorhexidine is that the presence of organic matter such as pus or scabs will limit yung kanilang effectivity. Yan. Next, we have iodophores. So this includes iodine, chlorine, etc. Iodine, let's focus on iodine since siya yung ginagamit natin in practice. So iodine, this is actually strong. So even in the presence of organic matter, pwede siyang mag-work pa rin. However, highly irritating siya, especially if hindi siya diluted. Next, we have hydrogen peroxide. Hydrogen peroxide, also, this is good pag meron tayong organic matter na present. However, certain bacterial species, especially yung mga may coagulase, this will limit the function of our hydrogen peroxide. For example, Staphylococcus, which are coagulase positive, kapag binuhusan nyo sila ng hydrogen peroxide, this will split into water and oxygen. Kaya nalilimit yung function nila. Next, we have sodium hypochlorite, yung Clorox natin. Do take note na sodium hypochlorite is recommended for disinfection of areas, especially sa clinics, kasi certain viral species hindi sila namamatay through alcohol alone. Ayun. So for microbial control, meron pa tayong antibiotics. However, hindi ko na siya in-include muna since maano naman natin siya sa pharmacology. Matatakal natin sila. So introduction lang, mga common. So we have the beta-lactam antibiotics, the tetracyclines, nitro-imidazole, etc. I included that sa handout natin. Andun na rin yung summarize na tawag dito. Summarize na mode of actions nila respectively. Ayun. I'll send a supplemental material later in terms sa sources naman ng ating mga antibiotics. Okay? So that concludes my lecture for today. Thank you for listening po.